stuff to go over, but I just wanted to hit all this complicated stuff one more time because I know at the end of the last chapter I was sort of pushed for time. So let's start over here. This is the renin, angiotensin, aldosterone system, right? And the key that I want to point out today is this top piece. So the body responds in many of the same ways if any of these things are true. So, you know, we talked last time and the time before about what happens when blood pressure is too low, you know, and we get that response of the heart and eventually the kidney to increase cardiac output by increasing, you know, some of these things, contractility, heart rate, all that. Well, what's interesting is the body pretty much responds the same if there's a decrease in blood pressure, a decrease in pH, oxygen, or an increase in CO2. Now, why is that? Well, this first piece, you know, blood pressure, really has to do with how well the heart is working and then how vasoconstricted the body is. But these other three pieces are all chemical indicators of metabolism. In other words, if the body's tissues are not getting enough blood flow, you're going to see this happen to the blood. You're going to see the pH go down, the oxygen go down, and the CO2 go up. Why? A busy tissue, you know, like think about a skeletal muscle. You know, you're doing anaerobic exercises, you know, so you're sprinting. And part of that process is the creation of acid. So busy tissues create acid. Acid lowers the pH. So that's the pH part. Oxygen. Busy tissues use up oxygen. So as oxygen levels fall in the blood, the kidney recognizes that as an inadequate blood flow, because that's usually what the cause is. You know, the oxygen concentration in the air doesn't change, but the busier we are, the more oxygen we consume. And then finally, if the CO2 is going up, that too is a sign that the cardiovascular system and the lungs are not keeping up with what the body's needs are, you know, which is to bring in oxygen and get rid of CO2. So the response to any of these four things is that renin, angiotensin, aldosterone system. So the kidney responds to this by releasing renin. Renin creates angiotensin 1. Angiotensin 1 is converted by the ACE enzyme into angiotensin 2. And then we get sort of two paths of results. One is an increase in total peripheral resistance. That's going to directly increase blood pressure. So that term. So angiotensin 2 directly increases blood pressure by increasing total peripheral resistance. So that's going to increase BP. But then the other role that angiotensin 2 plays is to trigger an increase in plasma volume, or how much blood there is circulating in the first place. So it does that by increasing aldosterone. So this system gets its name from these three chemicals, so renin, angiotensin, aldosterone. So why do I keep repeating myself about this? We're going to see this in two other chapters too. So it's not just a right now concern. It's also, you know, we're going to see this system at work in a couple of other places. So I just want you to sort of have seen it as simply as I can put it, which is like this. Now, the other response of the kidney to these conditions is to increase the number of red blood cells by releasing erythropoietin, right? So these are the long-term controls, or we'll say long-term responses. Because in the short term, the body's going to affect things it can affect immediately. So, for example, if the blood pressure gets too low, or the pH gets too low, or the oxygen level gets too low, the body's first going to increase heart rate, because that it can do real fast. Then it's going to increase contractility, so it's going to make the heart beat better, you know, contract more completely at the end of each contraction. So that's going to increase stroke volume. The body is going to increase venous return. It has some tricks for doing that, so it's going to do that. As venous return increases, end diastolic volume goes up, stroke volume goes up, cardiac output goes up. And then, so those are immediate changes. You know, heart rate, contractility, venous return. This takes a little longer, but it works better. So here again, when blood pressure is too low, when the pH is too low, oxygen is too low, CO2 is too low, the TPR is going to be increased. So what we see is basal constriction. So the vessels in the body all get a little smaller. That makes the resistance to blood flow a little higher, and the blood pressure goes up. So even though your book deals with these cases separately, you know, the case of blood pressure that's too low, the case of oxygen levels that are too low. The reality is the body's responding the same way to all of those things. And that's to increase what it can immediately and then eventually solve the problem with a longer term response. All right, so I added some things here just to make it more clear. Plasma volume and venous return are related to each other. As plasma volume goes up, venous return goes up. That makes end diastolic volume go up, right? So what is that saying? The more blood you have in the system, the more blood the heart gets before it contracts, the bigger it is, and therefore the greater the amount it can actually push out in a single contraction. The opposite's also true. As your venous or as your plasma volume goes down, you know, you've got a head of hemorrhage, let's say, your venous return goes down. That causes your end diastolic volume to go down and your cardiac output to go down. So plasma volume and venous return are related. But plasma volume is not the only thing that affects venous return. So even if your plasma volume stays the same, venous return can still change. So for example, you know, by the end of our class period, you guys will have some blood pooling in your feet because you've been sitting still for a long time. And then when you get up to walk down the hallway, your venous return will go up as the skeletal muscle pumps blood back up towards the heart. So even though plasma volume and venous return are related, there are more ways venous return can change than just plasma volume. Does that kind of make sense? Okay. So anything that makes venous return go up will make stroke volume go up and your cardiac output go up. So in lab, did you guys do the squat thing? Yeah. So that's another way to increase your venous return. Turns out when you do that, you compress your abdomen, and that compression pushes a ton of blood up towards your heart, pushes it up into your thorax. So you get this little spike of stroke volume, which makes your blood pressure go up. So it's just an example of venous return. And when you laid down for a long time and then stood up real fast, that's the opposite. Your venous return goes plonk, because as you stand up, all your blood goes to your feet. That makes your venous return fall. Your end diastolic volume then drops. That reduces your cardiac output, and you get a little low blood pressure, right? So, examples. The other piece of EDP that we talked about was filling time. That's related to heart rate. So as the heart beats faster and faster, it has less and less time to fill. So it ends up reducing EDV, stroke volume, and cardiac output. All right, so those are the EDV pieces we talked about. The ESV pieces, afterload, which I think is probably the most difficult to conceptualize or understand. So the simplest way of thinking of afterload is think of it as the diastolic blood pressure. The more pressure the heart has to pump against to move blood, the less well it's going to pump, the less well it's going to contract. So as your diastolic goes up, your end systolic volume goes up, and that makes your cardiac output go down. So there's that piece. Contractility affects ESV in a positive way. So when the sympathetic nervous system is trying to get the heart to increase cardiac output, it not only increases heart rate, but it increases contractility. So the heart gets smaller with each beat. It contracts harder, it squeezes harder. So that lowers your end systolic volume, subtract a smaller number, and you have a bigger number. So that increases cardiac output. All right. And then the last piece that affects both EDV and ESV is the Frank-Starling relationship, which is always true. And it says that as the end diastolic volume goes up, the end systolic volume goes down. So the more work the heart, a way to state this without terms like that is the more work the heart has to do, the more work the heart can do, is sort of the idea here. So end diastolic volume goes up, end systolic volume goes down, and that makes a big increase in stroke volume. So it makes a big increase in cardiac output. This relationship is part of how we can do that amazing exercise adaptation that we ended with last time. You know, how can we go from 6 liters per minute of cardiac output to 19 liters per minute? Part of it is this. At rest, our hearts are not working optimally. And that's fine because they don't need to. You know, they don't have as much to do. But as we get to moderate exercise, you know, think about a marathon runner pace, for example. The heart actually gets into its most efficient size because it's filling up a little bit more than it does when we're at rest, and that causes it to empty a little bit better. So one of the reasons that we can do that is this property of the heart. And like a synthetic or mechanical heart, you know, replacement, this is one of the barriers to that. Is it's very difficult to design a pump that can do this same thing. And it comes about because of how our hearts work, you know, with the sarcomere structure. All right, anyway, I depart. Frank-Starling Relationship. And then the last piece that turns cardiac output into blood pressure is total peripheral resistance. And it really just has 2 things it can do. We can have vasoconstriction or vasodilation. So in vasoconstriction, all the vessels, all the arteries in particular, get a little smaller. That increases the resistance, which increases the blood pressure. And the good thing about that is blood pressure is the energy for blood flow. And because it's blood flow, it's what it's all about, right? Actually moving blood through the system. So when our vasoconstriction increases, total peripheral resistance goes up, and that means that now there's more energy in the system for the capillaries to use to bring blood flow through the tissues that need it. So vasoconstriction and vasodilation then lowers total peripheral resistance. All right, so I thought we would review the hard stuff first so that I had no board space. All right, but let us go back and look through a bunch of the things that we've talked about. All right, slideshow. All right, so some review points.
I don't see that.
Huh?
I don't see anywhere where it's the exam review.
You will after we're done.
Okay.
Yeah, this is going to appear in the clicker question folder.
Okay.
Because, well, you'll see in a minute, but we're going to end with some clicker questions. So, but that's where you'll find this. Okay. Feels like forever ago, but we did start this semester with endocrine. And one of the first things we talked about in the semester was that there are 3 different classes of chemical messengers, right? There's the amino acid derivatives, the peptide hormones, and then the lipid derivatives. And they have different properties. The first 2 are water soluble. So they have to interact with receptors on the cell surface, right? The third one, the lipid derivatives, they can pass right through cell membranes. So they can have direct effects inside the cell. And we talked about some mechanisms too, which we'll get to in a minute. Review the types of cell-to-cell communication from your book, because I would hate for you to miss a question because you missed a vocabulary word, so to speak. So remember, there was endocrine, paracrine, neural, and direct communication. It's just one box that gives you the different names. So just spend 5 minutes and look at those so you don't get a word that's weird. We talked about mechanisms. How do chemical messengers make cells change their behavior? Because remember, ultimately, that's what the endocrine system is for, is so that a messenger can trigger a change in a cell's behavior, get it to do something different, get it to do something more or less. So we talked about second messengers. how, we had, if this is the cell surface and this is a receptor, when something occupies that receptor, it causes an increase of something inside the cell. The one we talked about was cyclic AMP. So why do we call it a second messenger system? This is the first messenger, right? And then this messenger makes this happen. So the first messenger triggers the formation of the second messenger now inside the cell, and then that makes a change in cell behavior, right? Second messenger systems are fast. That's why they evolved. So a cell or a hormone like epinephrine, for example, can make a change in a cell's behavior very quickly because nothing has to get made. The second messenger is not being created, it's just being modified. So they're fast. We talked about the lipid derivatives work typically inside the nucleus because they can pass right through the cell membrane, right? So they turn genes on and off. The lipid derivatives usually do that. And, gene transcription takes time. So while the second messengers are fast, the intranuclear or the turning genes on and off is quite a bit slower because you have to turn the gene on, it has to get transcribed, it has to get translated, all those steps have to happen. So the response is slower, but it can be more extreme. All right. And then the intracellular mechanism was sort of a hybrid between the two. Instead of the messenger making its effect from the cell membrane, it binds to a receptor, but then the whole complex is taken in. So the receptor and the messenger go inside, and then they make a change. So change the cell behavior. So don't spend a lot of time on the messengers. Like the book goes into too much detail on the second messenger system. If you recognize cyclic AMP as a second messenger, you're in good shape. But don't worry about G proteins and all that stuff. Okay. Hypothalamus, pituitary. Okay. So we talked about how the hypothalamus controls the endocrine system, and the pituitary is sort of the hardest working gland. So here's the brain up here, and then there's this stalk here, and the pituitary hangs down from underneath. So the hypothalamus releases chemicals, and they travel through a portal system that takes them to the pituitary. So the hypothalamus sends a signal to the pituitary, and then the pituitary releases all of those hormones that we talked about, which we're going to get to in a minute. So the connection there is the portal system, which is a venous system that takes blood from the hypothalamus to the pituitary. All right, we talked about the two parts of the pituitary, the neurohypophysis and the adenohypophysis. So essentially, the back part of the pituitary acts like a bunch of nerves. The front part acts like a gland that produces chemical messengers. So know which comes from what. So the neural hypophysis, or the neural part of the pituitary, produces oxytocin and ADH, and then the front part of the pituitary, the adenohypophysis, produces the other pituitary hormones. So FSH, LH, ACTH, TSH, GH, prolactin, and melanocyte stimulating hormone. Okay, so you will see them as both spelled out and their abbreviations, because the textbook publisher sort of uses both. So be familiar with both. Now I can tell you, FSH and LH we didn't talk very much about because they're reproductive system hormones, so we're going to talk about them later. But ACTH, we did talk about. TSH, we certainly talked about. Growth hormone, we talked about a little. Prolactin, and I said melanocyte stimulating hormone is sort of insignificant because we don't really know what it does. So those all come from the pituitary, these guys. So there are 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 hormones that come from the pituitary. All right, and then what do? Well, the hormones from the pituitary trigger the release of hormones from other glands. Okay, that's sort of its job. The hypothalamus tells the pituitary to do something. The pituitary then tells the target glands to do something, right? So, for example, aldosterone comes from the adrenal gland. T3, T4, those come from the thyroid gland. Cortisol also comes from the adrenal gland. It's released in response to ACTH. Somatomedins come from the liver in response to growth hormone. Insulin and glucagon come from the pancreas. They're not controlled by the pituitary, so insulin and glucagon are sort of on their own. The pancreas controls blood sugar by monitoring blood sugar itself. So insulin makes blood sugar go down, and glucagon makes blood sugar go up. We talked a little bit about glucose homeostasis, and you should review that because there is so much diabetes, particularly type 2, in our country today, that in your various classes and certifying exams, you're guaranteed some questions about glucose homeostasis because it's just a hot topic. So review insulin and glucagon. Calcium homeostasis is accomplished by calcitonin and parathyroid hormone. Parathyroid hormone increases calcium levels, calcitonin decreases them, right? And with calcium homeostasis, there's this little added complexity in the fact that the bones are involved. So calcium can be released from bones, calcium can be stored in bones. So when you look at that, be sure you include that bone piece. So for example, when parathyroid hormone goes up, the body's response is to try to increase calcium levels. One of the ways it does that is by dissolving calcium out of the bones. So parathyroid hormone over the long term makes bones weaker and weaker at the expense or for having a higher calcium level in the blood. All right, so that's that one. Erythropoietin, we just talked about a little bit earlier today, triggers the formation of red blood cells, and it's released by the kidney. So the kidney is in charge of determining how many red blood cells you have, and its method or its messenger is erythropoietin. Here again, erythropoietin has been synthesized, so we can give it as a medication, and it is being given as a medication to huge numbers of people. So anybody who's on chemotherapy, anybody who's on auto or immune therapy, anybody who's got a bad anemia like sickle cell disease, they're very likely to get erythropoietin. So it's very commonly used. And then, of course, epinephrine is an amino acid derivative, and it comes from the adrenal gland, from the adrenal medulla. Probably one of the most important hormones in the body because epinephrine is the principal sympathetic nervous system. I can't see it anymore, but sympathetic nervous system hormone. It's also a rescue drug. So if you ever see somebody dying of a heart attack in the emergency room, they're going to give epinephrine every 5 minutes until they call the code. And that's because epinephrine can keep you alive because it can stimulate the heart to return to its normal functioning. All right. So lots of hormones. Spend a little time with those. We talked about the stress response. It has 3 phases. The alarm phase, that's controlled principally by epinephrine, right? That's the fight or flight response. Adrenaline, that's epinephrine, is adrenaline. So the first phase, the alarm phase, optimizes the body for peak performance. You know, if you're going to fight or flee, you want to be able to fight as good as you can or flee as fast as you can. So that's the first phase. The second phase is the one that people usually have never heard of, and that's the resistance phase. When you think resistance phase, think cortisol, because cortisol plays a major role there. And the way I think of the resistance phase is once you have fought or fled the mountain lion, you are probably not in perfect health, but you still need to survive. So what cortisol does is it makes you resistant to death, is basically what the resistance is. So in that phase, you don't have to eat as much because cortisol is keeping your blood sugar levels high. You don't feel the injuries as much because the immune system is turned down. So it's cortisol driven, and it's to get you from escaping with your life to actually starting to heal, right? Now, the third phase only happens if the stressor doesn't go away, and that's the exhaustion phase. So all the hormones in the world are not going to keep the body alive, right? So if that stressor doesn't go away, then eventually the system starts to collapse. You know, you run out of available blood sugar, you run out of nutrients, the immune system starts to collapse. So in the exhaustion phase, the stress response kind of wears out, and it can't help anymore. So there's a nice 2-page spread in your book on the stress response. And I encourage you to look at it, even though we didn't get to spend very much time on it in class, because it came at the end of that chapter. All right, so here I've said it again. Blood glucose regulation, be sure you look at that. So what, why, and how. So what is involved in it, why, and how do those systems work, okay? And then calcium homeostasis, we already talked about that. All right, so then from there, we got into the blood, and we talked about what is blood made of. Plasma and formed elements, right? An important distinction. So you take blood out of your arm. If you pull off all the cells, what's left behind is plasma, okay? So plasma does not, is not inside of cells, it's outside of cells. And one of the most important parts of plasma is the proteins it contains. So we talked about albumin, we talked about the globulins, and then we talked about some of the inorganic stuff. So, you know, the sodium, chloride, calcium, those things. So plasma is proteins. The formed elements are red blood cells, white blood cells, and platelets, right? Anything with a cell membrane is a formed element. Then we talked about the life of a red cell, how they're created in the bone marrow. They're basically, they lose their nuclei before they enter the circulation. They're full of hemoglobin, and we talked a little bit about hemoglobin. You know, it's a 4-subunit protein. Each subunit has a heme molecule in it, and each heme molecule has an iron ion in the center that holds a molecule of oxygen as it travels around the body. So we talked about that. We talked about how iron is recycled, right? And we talked about as a red blood cell gets old and dies, and its pieces are recycled, a hard piece to recycle is that heme molecule that's part of hemoglobin. So you can kind of think of it as, lead. We put stuff out in our recycle bins, right? Some things are more easily recycled than others. And this heme molecule is like lead because it's very difficult to break. It's very hard to get rid of. So essentially what the best the body can do is it takes this heme molecule and it makes bilirubin out of it. And then bilirubin is ejected into the GI tract by the liver. So have looked at that because the iron recycling piece and the bilirubin piece both come up clinically. So you have to know a little bit about that. The 5 types of white blood cells, and each one of those had a claim to fame, so to speak. So the neutrophils, their remembered things are they're first on the scene, and that's because the bone marrow can produce them very quickly. They're very active against bacteria. So in a bacterial infection, you have mostly neutrophils doing the work. And then they're phagocytic. So they do their job by gobbling things up. So that's the neutrophils. The eosinophils, I told you to think parasites and allergies. So eosinophils are related to those 2 things. Basophils, they release histamine, and they're dark purple, so you can't see their nucleus. That was the things to remember there. So those are the granulocytes, neutrophils, eosinophils, basophils. the other two white blood cells are the monocytes and the lymphocytes. Monocytes are baby macrophages. Macrophages are great big huge cells that live in the tissues. So monocytes are on their way to become these giant cells that help defend us from things in our environment. And then the lymphocytes, I said, are the boring cells that do exciting things. And in the next chapter that we get into next week, we'll start seeing what the lymphocytes do. But for now, they're tiny little cells. They have a great big nucleus, but not much else. And that they look boring, but as we'll see, they do exciting things. So the different types of white blood cells. Okay. We talked about hemostasis and the three phases of it. So the vascular phase, the platelet phase, and the coagulation phase. In the vascular phase, the vessel responds to the injury actively. So like it gets smaller is one thing that it does to try to keep blood from flowing out of it. The endothelial cells that line the vessel also respond to the injury by shrinking. And then as they shrink, that's what triggers the platelets to start sticking to that region of damage. So when platelets stick to something, they become sticky to other platelets. So you end up with this sort of pylon of platelets all stuck together. That's the platelet plug. And then the platelet plug helps to trigger the coagulation pathway that we talked about. And the coagulation pathway was very complex, as we saw, but I told you that all I wanted you to know was sort of this. All right, so there's an extrinsic pathway and an intrinsic pathway, right? Both of those require calcium. So for sure, no, blood doesn't clot without calcium, right? And it clots too much with too much calcium. So calcium and blood clotting should be forever associated in your mind. All right, but that big giant equation, all I wanted you to know was both the intrinsic and extrinsic pathway result in prothrombinase. Prothrombinase turns prothrombin into thrombin. Now, thrombin, why that name? A thrombus is a blood clot, particularly a blood clot that is stuck to a wall, not moving through the system. So prothrombinase turns prothrombin into thrombin, and then thrombin turns fibrinogen into fibrin. Fibrin makes a clot. Okay? So that was all I wanted you to know about that big complicated cascade. Is prothrombinase, prothrombin becomes thrombin, fibrinogen becomes fibrin, fibrin makes a blood clot. Now what about those last three words? Well, we have to get rid of a clot sometimes too. So plasminogen becomes plasmin, and then plasmin destroys the clot, right? The enzyme that makes this happen, the one I want you to know, is called TPA. So that's tissue plasminogen activator. All right, so a little review of coagulation. All right, okay. So then, then we talked about vessels. My review is out of order, but for the heart, we went through the flow of blood, right? How blood comes back to the heart, to the right atrium, from the right atrium it goes to the right ventricle, and so on. You know, we did that step by step on the board. Be sure you know that, because you're going to get questions about the flow of blood through the heart. It's just, you have to know that. And the way we arranged it on the board is that then what valves does it pass through, or what arteries does it pass through? So do review that. The differences between the left and the right side of the heart, and why. You know, the left side is a high resistance, long distance circuit. So the ventricle is very strong and thick, whereas the right side of the heart, a very low resistance pathway. So the right side of the heart is sort of thinner and less muscular. Review the branches of the coronary arteries. So on the left, you have the circumflex, and you have the left anterior descending. Then the right coronary artery has the posterior descending, and the marginal arteries that come out. So review that anatomy. The conduction system, SA node, AV node, bundle of His, bundle branches, Purkinje fibers, right? That should sound a little familiar. EKG, P, QRS, and T, and what each of those mean, right? I'm not going to ask you about abnormal EKGs, though, on the exam, because we talked nothing about that. All I want you to know is be able to recognize a P, a QRS, and a T, and know what each of those things represents, right? And that's atrial depolarization, ventricular depolarization, or ventricular repolarization. Okay. So the heart has 4 valves, and we talked about them. You should know when the valves open and when the valves close, right? So when do the AV valves open? When do the AV valves close? And the best way to think of it is in terms of pressures. You know, we looked at that chart that had pressure on the left and then time on the X. So when one line crossed another, a valve opened or a valve closed, right? So do review that. The heart sounds, you know, there's 2 of them, S1 and S2. I won't ask you about S3 or S4 because those are technically abnormal. But an important distinction is there are 2 heart sounds, but there are 4 valves doing things, right? So the first heart sound is 2 valves closing. The second heart sound is 2 valves closing. So valves only make a sound when they close. And that's because just like slamming a door makes a sound, but throwing a door open doesn't necessarily make a sound, it's the same idea. When the valves close, they slam shut, so they make a noise. So S1 and S2 are the 2 heart sounds. And then that last bit, which looks simple, that's what we were reviewing earlier. So cardiac output, stroke volume, all those terms that we talked about. Okay. And then finally, the blood vessels and blood pressure. Okay, so review the arterial system. So I gave you a study slide with arteries and veins on it. You may see a similar diagram on the exam. So, you know, you want to have those in your head, even though I know it's sort of pure memorization. Did you get to work with any vessel diagrams in lab? No. Some of you did, some of you didn't. Did you?
The vessel diagram?
We have this whole packet that we have to like.
Yeah.
Excellent. Good. Super boring, I'm sure. But.
I don't know. Like, the pictures weren't that great though.
Yeah, it's hard to get good pictures for these things, but it's the exercise is good.
Yeah.
So I'm glad that you were able to look at some of those. Do take 5-10 minutes at least and review the cerebral arterial circle. It's one of my favorite arterial spots in the body. So I tend to pick questions about it, at least that's what I've been told. So do have looked at those, so you can get that question. There's not going to be 10 questions on it or whatever, but there'll be at least one. The venous system, normally, for every artery, there's a vein with the same name. At least that's the common pattern. The only difference is in the arms and legs, there is then a second vein as well. One that goes deep and one that goes over the skin, just superficial under the skin. So you have some different ones there. We talked about the differences between arteries and veins, how they look under the microscope, how their walls are different. The tunica media is different between the two. The endothelial cells are different. Arteries, the endothelial cells are sort of ruffled, and then veins, they're nice and smooth. We talked about properties of the cardiovascular system. Can I erase this? Nobody complaining? Okay. And the one I want to just refresh your memory about is the roller coaster piece. All right, so if this is pressure on this side, and then along here is, we're traveling through the tract. So we go from aorta back to vena cava. the pressure is always decreasing. It's like that. So we talked about it as a roller coaster. The capillaries are here in the middle, so caps. So the area of the cardiovascular system with the lowest pressure is the veins right before the heart. Highest pressure, aorta, capillaries are in the middle. Capillaries are not the lowest pressure area, right? So we talked about it as a roller coaster. The heart pumps the blood or pressurizes the blood, and then the blood flows through the system and then back to the heart where it gets pressurized again. All right? So there's that piece. Okay. And then we talked about the interstates and side streets, that the larger vessels, there are fewer of them, just like interstates, but they're higher speed and they're larger. Same idea in the cardiovascular system is the larger vessels, there are fewer of them, but the blood moves through them faster and they're larger. Right. And then we, of course, talked about capillary exchange, where we have those two opposing forces, hydrostatic pressure pushing fluid out and colloid osmotic pressure pulling fluid back in, and how they don't quite balance. So in all the capillary beds, a little more fluid leaves than comes back in. So there would be extra fluid all the time in the body if it wasn't for the second vessel system that we're going to talk about in the next chapter, the lymphatic system. All right. And then we talked about blood pressure control, which we were reviewing earlier, and regulation. So autoregulation is the principal way that blood flow is regulated. So what does that mean? All of the capillary beds in the body can regulate themselves. In other words, if a tissue is busy, the capillary beds in that tissue will be open for blood flow without the brain having to be involved, without any of those systems over there having to be involved. So that's autoregulation. The capillary beds regulate themselves. But when that fails, in other words, when there isn't enough blood pressure to provide for all the capillary beds that need blood flow at that time, then we get into the neural and hormonal systems. So in our chart here, the neural piece, that's the sympathetic and parasympathetic nervous system. So we saw that it can increase and decrease heart rate. It can increase and decrease total peripheral resistance through basal constriction or vasodilation. The neural can affect contractility, so it can make the heart beat harder, which also increases blood pressure. And then the hormonal piece, that's this piece. That's the renin, angiotensin, aldosterone system, is the hormonal control of blood pressure and blood volume. All right. Okay. And onward. Okay. So those were my review points.
Could you go back to the one that had like the arteries and stuff on it? I just didn't know if I missed anything.
Which one? This one?
No, the one before.
That one?
Can you just leave it up there for just a little bit? I
This one?
Get the bottom.
Well, I am going to post it.
Oh, it is on there.
Yeah, I mean, it'll be in the clicker question folder.
Okay.
Oh, you're talking about all these terms?
Mm-hmm.
Yes, that's why I typed them all out, is so that you'd have them. Yeah, I'm going to post this whole thing.
All right.
All right. Okay, so we have not very many minutes, but let us just do some questions. All right. I'm going to skip some of these. Okay. How about this one? Which of the following plasma proteins is most abundant? So you should get the question now. Is that erasable, or do you guys want to take pictures of it later? Okay, I'll leave it. 1, 2, 3, 4, 5, 6. All right, last person, jump in there. right, good. We have some variation, which means it's a good question I've talked about. Okay, so plasma proteins are proteins in the plasma. Okay, plasma is the part of the blood outside of red cells, or outside of cells. So, of those, E, it can't be E, because hemoglobin is found only inside of red blood cells. And for a good reason. Hemoglobin, when it's in the plasma, it wreaks havoc. It damages the kidney, it damages vessels, capillary beds. So the only place you find hemoglobin is inside of red blood cells. So if we throw that one out, which of those is the most abundant? A, albumin. And albumin is produced by the liver, and its primary function is to create that osmotic pressure that we talked about in capillary exchange, the blood colloid osmotic pressure. Most of that is because of albumin. So in treating patients sometimes, if we don't want to give them blood, we can give them albumin instead, and it can perform some of the same functions. Of course, it can't carry oxygen, but it can help to keep water in the vessels. All right. Here we go. We're not going to do that one either. Here's a good one. During platelet plug formation, what happens? And attached to this or next to it on Angel, there's a whole bunch of questions here. There's like 40 questions. And there'll be a separate file that has the questions and then the answers at the bottom, so that you have those to study from. I've stumped you, I guess. I got one answer so far. All right, jump in there. Okay, good. A is the best answer. Platelets can either be sticky or not sticky. And when platelets are doing their job, they're sticky. In other words, when they're helping to seal a hole in the vasculature, they're sticky. What do they stick to? They stick to anything that's abnormal in the vessel. So that's damaged endothelial cells, the material underneath the endothelial cells, and to other platelets that have already gotten sticky. So we call that platelet adhesion. And, we've all heard about, take a baby aspirin once a day, right? And that's supposed to reduce your risk of stroke if you're over 50. The reason for that is aspirin keeps platelets from getting sticky. So it makes it harder to make a clot, which is sometimes a good thing, sometimes a bad thing. But that's the effect is on platelet adhesion. All right, here we go. Blood in the pulmonary veins returns to which part of the heart? And then we'll call it a day after this one. All right, okay, good. Most of you got that. So blood comes back from the lungs to the pulmonary veins into the left atrium. There are 4 pulmonary veins, top and bottom, left and right. All right, that's it for the day.